Hola, Team Bio. Okay, so today's screencast is going to be on DNA fingerprinting. This is the process that's used to solve murder mysteries, and it is the process that you're going to have to know and understand for our culminating experience next week. So I'm going to explain here. It's going to be a relatively long screencast because this is a long and complicated process. But I don't think it will be too terribly arduous. It's actually pretty cool. So, as I said before, DNA fingerprinting is the way in which murder mysteries or just crimes are solved, and it allows DNA found at a crime scene to be matched against potential suspects of the crime. And it can be broken up into two parts. The first part is called PCR, and the second part is called gel electrophoresis. I think I spelled that right. Okay, so composed of two parts, and the first part is PCR, and we're going to talk about that first. Surprise, surprise. So, PCR. It stands for polymerase chain reaction. And as you know, to build a polymer of something is to put many mers together, or many things together. And in this case, we are going to polymerize DNA. And it's a chain reaction because it happens again and again and again and again. The guy who invented PCR is this scientist named Kary Mullis. And interesting story, he was actually high on drugs when he came up with the idea for PCR. If you want to learn more about how he thought of it, I suggest doing some online research. It's actually kind of interesting. Okay, I'm going to erase that. Okay, so PCR, polymerase chain reaction, is a way to magnify DNA, and this is the way that it works. Okay, so if you have a strand of DNA, like so, and you take that DNA and you, first of all, you heat it up. You add some heat. And the heat causes the strands of DNA to separate from one another. Because remember, the bond that holds the bases together in the middle, those are hydrogen bonds, and they are relatively weak bonds. So heating up the strand of DNA separates the two sides, and however, the phosphodiester bonds, the covalent bonds that hold the bases to the sugar and the phosphate group together and the phosphate to the next nucleotide, are all covalent and they stay together. So this is a way to denature the DNA. We heat it up a little bit. And then, in this mixture, we add a few things. We add something called an RNA primer. So in your DNA replication videos, some of you included the fact or mentioned, oh, actually none of you did because it was stop action. Well, anyway, a RNA primer is a necessary component of basically starting DNA replication. And DNA polymerase, oh, this is terrible because it doesn't actually line up. DNA polymerase can't start, I'm just going to do it over again. Terrible. Okay, DNA polymerase can't start synthesizing a strand of DNA without an RNA primer already there. So you have primers in the solution as well as a bunch of free nucleotides in the solution, and a polymerase, a DNA polymerase that's actually a heat-resistant polymerase that's in the solution as well. It's called Taq polymerase. It's found in bacteria that live in hydrothermal vents, so deep within the ocean in very, very hot locations. So as you know, these polymerases are proteins, and heat has the potential to denature proteins, but these proteins, because they're found in these bacteria that live in this really high temperature, don't denature in the heat. So You add all these things together, and guess what? DNA replication will take place. These Taq polymerases will zip along these strands of DNA, and they will copy the DNA. So after one round of replication, you double the amount of DNA. And so it will look like this. Boom, boom, boom. So if you start out with one piece, by the end of one round of replication, you have two pieces. And then you add heat again. So heat here and heat here. Remember, this is all taking place in the same test tube. Spelled that wrong. Heat. And again, these will break apart, and all the same pieces that were present in the test tube before, our primers, our bases, and our Taq polymerase will all jump on here, and so we'll get another round of replication, and voila. So the amount of DNA doubles after every cycle. So if we were to look at a graph of the amount of DNA over time, we'd see it looks like this. We get a lot of DNA after just a few cycles of heating and cooling. So yeah, so ultimately you get a ton of DNA. And this is important because we're going to try and analyze this DNA, but if we're at a crime scene, potentially, let's say we have this is a terrible t-shirt. This is, terrible, really bad t-shirt. But imagine we just have a teeny little bit of blood on this t-shirt. Or here's the suspect's blood's all over the t-shirt, but we've got another little bit of blood that's not the, so I should say the victim. This would be the victim's blood. A lot of victim blood, and then a little bit of blood right here. This is the suspect. And so by amplifying the amount of DNA suspect sample that we have, we can actually do something with it. We can analyze it. So PCR is always run as a way to collect a bunch of DNA. Okay, that's not that complicated, so let's get on to the next bit. So this is PCR, and the next piece is gel electrophoresis. Okay, so the way gel electrophoresis works, I'm going to go back for a second. So the point of this was to magnify the DNA, or increase the amount of DNA that we have to analyze. But just this DNA doesn't really tell us anything. So we've magnified this piece of DNA, we now have a lot of it, but we don't know who it is, who it belongs to. So the gel electrophoresis bit allows us to identify whoever's DNA it actually is. Okay, and this is the way it works. Before we even begin gel electrophoresis, we have to do something to the DNA. And so this is called a restriction enzyme digest. So restriction enzymes cut DNA, but they're called restriction enzymes because they're restricted in the place that they can cut the DNA. So for example, one restriction enzyme is called HindE3, and this restriction enzyme only cuts in a certain place. It only cuts when it sees a sequence of DNA that looks like this: A, G, C, T, T. An interesting thing about restriction enzymes is they often cut at palindromes. And a palindrome is something that's the same backwards as it is forward. So for example, the word race car. As you can see here, R-A-C-E-C-A-R, this is the same if you read it this way as well as if you read it this way. So HindE3 actually cuts at a DNA palindrome as well. But the way we talk about DNA palindromes is a little bit different than the way written words are palindromes. This is a palindrome because the DNA this way on this strand is the same as the DNA on the complementary strand going in the other direction. So if we were to fill in this, this would be A, G, C, T, T. So you see here, their palindromic sequences top and bottom. Okay, so Hindi 3, the way that it cuts is like this. So ultimately, on one end, we'll have a strand of DNA with an A, and then another strand of DNA, and there'll be a T, T, C, G, A here. And then our other side will look like this. There'll be an A here, G, C, T, T, more DNA, and then an A down here. So we cut the DNA just like that. Okay, so that's what restriction enzymes do, and you might be asking, one second, why is it important that we cut the DNA like this? And the reason that it's important is because people have different restriction enzyme sites. So let's say we have a couple different samples of DNA here, and so if this is suspect 1, and this is suspect 2, and this is crime scene DNA, we are going to expose these pieces of DNA to this restriction enzyme, Hindi 3. And wherever it sees this site right here, it's going to cut. So let's say for suspect 1, that happens twice. Happens once here, and suspect 2, that happens once, and the crime scene DNA, that also happens once. So we're going to end up, ultimately, this guy, we're going to see 3 fragments that are all the same size. And from this guy, we're going to see 2 fragments that is 1 short and 1 long. And from this guy, we're also going to see 2 fragments that's 1 short and 1 long. So right away, you guys can already tell, oh look, suspect 2 is actually the one who committed the crime, or at least left their DNA at the scene of the crime. And that's all well and good, woohoo, you're right. But this is all happening, remember, in a test tube, and we can't actually see the different restriction enzyme fragments that come out of this. So we need some way to visualize it, and that's where gel electrophoresis comes into play. So here's what happens next. What you do is you create a gel. Here's this gel, and the gel is going to have little wells in it that are like, oh wait, they're like little divots in the, oh, I'm totally drawing this terribly. Little sections of the gel that are missing. Oh, I've only given myself space for 2 here, but in reality there would be 3. So again, if we're running 3 samples, we're actually going to have 3 spaces in the gel. But I'm just going to draw 2 here. And you take these little wells, they're called, and you squirt some DNA into them. This one, and then this one. And the gel is a matrix through which the DNA is going to run. And how do I want to describe it? Okay, so imagine that you are in a crowd of people. All these little dots are people. And you want to get from one side of the crowd, you're up here in the red, to the other side of the crowd. This might be the journey that you take. And you end up over here, right? But imagine that you're in this crowd and you have to hold hands with someone else. And they're holding hands with someone else and someone else. It's going to be a lot harder for this whole chain of you guys to all move through this crowd of other people. because you're all holding hands. The way that DNA moves through the gel is very similar. So ultimately here, if you're just one person, you're going to move through a crowd of people a lot faster than if you are a long chain of people. The same can be said for strands of DNA. If you are a little short strand of DNA going through a gel now, you are going to go a lot faster through the gel because you're not impeded by all the little gel particles than if you are a long strand of DNA. So what we're going to do is we're going to run DNA through the gel, and these pieces are actually going to separate out by size. So let's look at our gel top down now. This is our gel, and here are our wells with our DNA in them. The DNA pieces are going to run down the gel, and the smallest ones are going to end up here, and the biggest ones are going to be back here. Because it's easier, again, for the small pieces of DNA to move through the matrix of the gel. But this DNA is actually not going to migrate unless you do something to it. And this is what you have to do. You have to hook it up to, this is a plug, can't you tell? An electrical circuit. And DNA, the molecule, is negatively charged. So if you have a positive electrode down here and a negative electrode down here, the DNA is going to move away from the negative electrode because it is negatively charged itself towards the positive electrode down here. So all these DNA are going to leave the empty space of the well and enter the gel. And again, the smallest pieces are going to migrate the fastest, but they're all going to be moving towards this positive piece. Okay, so now if we look back on what we saw here, let's label our wells here again. This is suspect 1, this is suspect 2, and this is crime scene DNA. Suspect 1, we created 3 small fragments, and they were all the same size. So that's going to show up at the gel as a band down here. And suspect 2, we created 1 small fragment and 1 big fragment. The small fragment was the same size as this one, and the big fragment is going to end up back here. And then the crime scene DNA is also going to look like this, the same pattern, because 1 big piece was created and 1 small piece in the restriction enzyme digest. Okay, so, but still, we've created this DNA, but you can't actually just see it on the gel. What you need to do in order to see it is hybridize it with a radioactive probe. So what you're going to do is you're going to create a probe for a known sequence of DNA, and then you are going to expose that to your gel, and the probe is going to bind to whatever sequence it is, and then you'll see a different pattern emerge. So here, let's imagine that the sequence that we used for our probe matched somewhere up here. So this is where the probe would bind to, because the probe is specific for 1 segment of the DNA. And so you see here that this probe binds to what ends up being a small fragment in suspect 1, but is a big fragment is in suspect 2 and the crime scene DNA. So we see it here as we only see signal in this location in both suspect 2 and the crime scene DNA, but over here with suspect 1. And remember, the way we're going to look for this probe is hold up this whole piece of gel electrophoresis to a photographic plate that's going to be exposed. So we're going to see light here and here and here. And that's how we know that it is indeed suspect 2 whose DNA we found at the scene of the crime. Okay, I think that is it. If you have any questions about this, we're going to be talking about it in class on Thursday a little bit to help set us up to do the murder mystery during our culminating experience slot. Okay, that is all.